Welcome to Science Hangout from Science Mission. This is our third Science Hangout. We have today Dr. Atsuo Sasaki, Assistant Professor of Medicine and Cancer Biology from University of Cincinnati, Ohio with us. Dr. Sasaki worked in Dr. Akihiko Yoshimura's lab at Kurume Medical School for his PhD. Dr. Sasaki later joined Dr. Richard Fertel's laboratory in University of California, San Diego, where he worked on to understand chemotaxis. Dr. Sasaki then joined Louis Cantley's lab at Harvard Medical School, where he investigated post-translational regulation of KRAS, one of the most frequently mutated oncogenes. He joined University of Cincinnati as a tenure-track assistant professor about 3.5 years ago. His recent seminal work involved identification of GTP sensor in the regulation of metabolism and tumorigenesis. Welcome, Dr. Sasaki. How did you start your project?
Thank you, Dr. Pai, for inviting me and the nice introduction. Hello, I'm Atsuo Sasaki. Our lab focuses on GTP metabolisms. We investigate how cells regulate GTP energy level and how cells detect GTP energy levels. I came to this GTP project thanks for the three fortunate things. Number one, as Dr. Pai introduced, I have studied for over 15 years on GTP binding protein called RAS. I know what is GTP and its importance in signaling. Number two, when I was a postdoc in Dr. Louis Cantley's lab in the Harvard Medical School, I observed myself that GTP level changed in one cell type to the other. Also, the ratio of GTP to GDP changes. I was shocked because in general, GTP is believed to be always high and consistent in the cell. Something happening. Number three, and the most important thing is that I have excellent friends who take my crazy idea seriously and continuously provides feedback and encouragement and supports. So these three fortunate project brought me to build the hypothesis that cells has GTP energy sensor that can translate GTP level into signaling for cell function. This is how our GTP journey has started. So the first challenge is that how to find GTP energy sensor, which we don't know anything about it. We don't know whether it exists or not even. From the 20,000 protein species in our cells. So we assumed that GTP sensors would bind to GTP. So we set up proteomic screening using GTP or ATP conjugated agarose. These agarose are mixed with cell lysate and washed and eluted in several conditions to identify GTP binding proteins. After mass spectrometry, we have found very interesting proteins named phosphatidylinosyl 5-phosphate 4 kinase, PI5P4 kinase beta. This kinase has a strong link to tumor as well as metabolisms. Knockout mice show hyperinsulin sensitivity and resist high-fat diet-induced obesity. Quite interesting phenotype. The knockout mice also shows a genetic link to the tumor. It's embryonic lethal when p53 knockout is co-deleted, and the duration of PI5P4 kinase activity in p53 knockout, which normally die within 10 months due to lymphomagenesis, suppress the tumorigenesis. PI5P4 kinase regulate two signaling lipid. So PI5P and PI45P2 are second messenger lipid. PI5P4 kinase phosphorylate PI5P to generate PI45P2, which is a substrate of PI345P3. While PI45P2 is mostly produced by this right wing, which is starting from PI4P, then this enzyme make PI45P2. The right wing is well-known pathway, but the one we have identified, this kinase is mysterious one. While it has been shown to use ATP as substrate, but we had the thought, since we have identified this kinase in GTP fraction, so we wanted to know whether this kinase could use GTP as substrate. We set up in vitro kinase assay. Recombinant PI5P4 kinase is incubated with radio-labeled ATP or GTP, and the product is analyzed by thin layer chromatography analysis. PI5P4 kinase use ATP as reported to generate PI45P2, and shocker is that PI5P4 kinase use GTP as preferred stronger substrate. Among the 500 kinases, PI5P4 kinase beta showed this very unique function that showed preferential GTP utilization in the kinase reaction. So we decided to take a biochemical analysis. This is an in vitro kinase assay. The ATP-dependent kinase activity is saturated around 1 millimolar of ATP. So in the cell, ATP level is something between 1 millimolar to 5 millimolar. So this kinase is saturated among these ATP concentration in the cell. GTP-dependent kinase activity dramatically increased up to 1 millimolar, and the kinase activity is always higher than that of ATP-dependent kinase activity. Importantly, this is the range for GTP cell concentration in the cell. Next, we did the competition experiment. This is the GTP-dependent kinase activity and we at excess amount of cold ATP, 1 millimolar and 2 millimolar. Even in this condition, PFIP4 kinase beta can use GTP to making PI45P2. These biochemical data strongly suggest that PFIP4 kinase beta can use GTP even in the cellular setting. However, the proof of in vivo significance and in vivo evidence is very challenging, since this kinase has a multiple function. It can use ATP and it can use GTP. And the current approach to modulate protein is not applicable for our purpose, since knocking out or knocking down of PFIP4 kinase take this ATP and the GTP dependent activity away. So we are stuck at this point. The breakthrough is coming from my friends. Dr. Ko Takeuchi, whom I met in Boston 2005, now in ICE Institute in Japan, pointed out that this project would need high-resolution structural analysis. Dr. Takeuchi then contacted with Dr. Toshiya Senda, who is a leading researcher in structural biology field. And Dr. Senda has formed the team for this project, Dr. Miki Senda and Dr. Yuhao Ro. So together, we tried to make X-ray structure of PFIP4 kinase bound with GTP. And then we realized that this enzyme is really difficult one to make that co-crystal. So, however, this incredible team didn't give up, and they tried multiple setting and condition, then even come up with a new technology to overcome this issue. So Dr. Senda and Dr. Takeuchi together identifies or develops a new method for crystallography, which enables us to have the PFIP4 kinase beta bound with GTP as well as ATP analogs. So PFIP4 kinase beta forms the beautiful dimer, which making the wide hydrophobic surface which attached to the membrane. And in the kinase pocket, we found GTP or ATP analogs are spot in. In the deep end of the catalytic pocket, ATP analog forms a 2 hydrogen bonding with PFIP4 kinase beta, while GTP analog form 4 hydrogen bonding, clearly explaining why this kinase prefer to bind and utilize GTP at atomic level. And this crystal structure pointed out which residues is critical for GTP dependent activity. With this structural information, we have made some mutant which shows a similar ATP dependent kinase activity to wild type. However, this mutant decreased the GTP-dependent kinase activity to the ATP-dependent kinase activity level. So this would be ideal probe to test the in vivo significance and the function of GTP-dependent kinase activity of PFIP4 kinase beta. So first, we used wild-type PFIP4 kinase beta, then expressed into PFIP4 kinase beta knockout methods. This is reconstitution assay. Then we used the mycophenolic acid, which reduced the intracellular GTP concentration to this level. Under this condition, the substrate of PFIP4 kinase, PFIP, is accumulated, suggesting that PFIP4 kinase beta is inhibited by cellular GTP reduction. Then we expressed mutant PFIP4 kinase beta. The mutant shows already higher PFIP level, and when GTP level is reduced, this PFIP is not increased, suggesting that in the cell, PFIP4 kinase beta use GTP as substrate. Then we wanted to know the cellular function of this GTP-dependent kinase activity. We tested the cell proliferation rate between wild type and the mutant, and actually there is not big difference. So under normal condition, GTP-dependent kinase activity seems not important. However, when we place the cells into soft agar, which is the assay to assess cellular anchorage independent growth activity, which lots of tumor acquires for their growth. So in this assay, mutant cell shows a reduced colony formation, so suggesting the PFIP4 kinase is important for transformation process. When we inject it into noodle mice of these cells, there's dramatic difference. Wild-type cells form tumors. However, we didn't see mutant cells forming tumor. These data show that GTP-dependent kinase activity or GTP sensing activity of PFIP4 kinase is critical for tumorigenesis. Based on this result, we think that PFIP4 kinase is a GTP energy sensor that can detect the change of cellular GTP levels, then convert it into phosphoinositide signaling pathway to regulate tumorigenesis. So I would like to thank a lot of people. I would need 10 slides to thank all of them. So I put just one slide for here. So this finding couldn't be done any of the person who give us advice, support, and encouragement. I would like to especially thank Dr. Ko Takeuchi and Dr. Toshiya Senda for their continuous and robust collaboration and with their passion to get through all of the difficulties to get this point. And thank you so much for listening.
Thank you, Dr. Sasaki. It's an interesting work. So I have a question. What is the most difficult part of this GTP sensor project?
I see. Yeah. Thank you, Dr. Pai. Well, I think we have a lot of difficulties to get to this point. And number one is to kick it off. This is totally new direction from what I have done. So I need courage to get into this space. So, but thanks for my friends and my family and my supporter. So I could step in to this very exciting project. And the second one is, as I mentioned, the structural biology. We had really struggle. The standard method basically wouldn't work for crystallize this enzyme with nucleotide. So, but the incredible team led by Dr. Senda made it through. I really thank you for their passion, and I appreciate their expertise. And the third one is the funding. Since concept is even not exist previously, we had a difficult time to convey what's a GTP energy sensor and what's the importance, potential importance in human disease. So we couldn't get the funding initially, but luckily we had the pilot funding, which enables us to develop this project.
That is interesting.
Thank you.
Are you looking for a grad student or postdoc in your lab?
Yes. We have a ongoing project for PI5P4 kinase inhibitor for therapeutic application. And also we are working on knocking mice and also cells.
Okay.
So there are lots of exciting projects which we want to further develop. And we are looking for a highly motivated and energetic graduate student and a postdoc.
Okay, that is interesting. So what is the best way to contact you? Email or any other way to get to you, like send the resume or so?
Yeah, that would help. Yeah. My door is always open. So email or just calling or chatting would be great.
Okay. Thank you so much, Professor Sasaki. Thanks for joining the Science Hangout and taking time from your busy schedule. We really appreciate your support, and we would like to see more of your publications and work in some prominent journals.
Wow.
And
thank you, Dr. Pai, for giving us this great opportunity to share our work. Thank you.
Thank you so much. So hopefully we will see you again on Science Hangout once you have some good data and publications.
Sounds good.
Thank you again. Bye.
Bye. Thank you.